So I'm the next speaker, as you guessed. And I'm based at Blood Systems Research Institute. It's a small to medium-sized research institute affiliated with UCSF, and we're based at a blood bank, so we look at things with a slightly different angle from everybody else, I think. We're thinking of the safety of the blood supply, and there's, since the HIV days and hepatitis days, there's been a lot of focus on infectious diseases in blood banking, and so that's the angle we come from. And West Nile virus was introduced into the US in 1999 and then spread sequentially over, got here in about 2004. And before they developed a screening test for it, there were quite a few transfusion transmissions of West Nile virus. And it tends to cause problems in blood recipients because they may be immune suppressed and do worse with the virus. So I'm going to talk a little about the clinical disease and then tell you some of the work we've done and really looking at it from an immunologist perspective, so not as much straight up virology. But this is West Nile virus. It was isolated from a patient in the West Nile region in Uganda in 1937. It's an arthropod-borne virus, mosquito-borne. And it's part of a family of dengue, yellow fever, Japanese encephalitis virus. They're all fairly closely related. And it has an organization with a capsid membrane envelope. There's a protease and helicase and RNA-dependent polymerase and methyltransferase. So it has all these usual viral proteins. And as immunologists, we just look at them as, does it react against them or not? Does our immune system react? And this is what happens with West Nile virus. We're dead-end hosts, and horses are dead-end hosts. There's a vaccine for horses, and it works reasonably well as far as I know. There's no approved vaccine for humans, and it's kind of a tough economic case, I think, to make because, as you'll see, most humans don't get severely symptomatic disease. But what I think is important about understanding this transmission cycle is that the birds get fairly high viral load, so they'll get 10 to the 9th copies per ml, and particularly the corvids. Crows, so when it ripped through St. Louis, about 90% of the crows died. And then you have cycles of them coming back, and we'll see some of the epidemiology as I go over that in the next few minutes. But what's important about this to understand for the virology, from a virologist's perspective, is what the virus is doing is going back and forth and adapting to the mosquitoes and to the birds, and because we're not part of the cycle, I think the adaptation to humans is really not relevant. So Eric Delward, who's at BSRI, looked at this with Belinda Herring, and they looked at the sequences and how much did they change. And if you put HIV here, it would look dramatically different, a lot more variability. So there's really not that much. These are sort of 1 or 2 nucleotide changes, and a lot of them were even synonymous mutations. So not a lot of variation happening over several years of the epidemic as it spread across the US. So it's not a highly variable virus. This is the envelope region here. And the virology, in terms of explaining who becomes symptomatic and who doesn't, it's probably not the virus that's the main determinant of that. So it really must be something to do with how we interact with that virus. Is how we think about it. So in terms of diseases, there are a lot of bad diseases out there, and we're hearing about some of them today. How does West Nile virus rate? Is it sort of wimpy and doesn't do a whole lot? And this actually looks like one of my friends, his head does, so I sent it to him in an email, and he didn't like it much. He's an orthopedic surgeon. They just can't take those jokes. Or is it a big gorilla? And that really depends on who you are. So like many things, as you get older, some things get better as you get older, but some things get worse. So if you're over 65, you're about 15 to 20 times more likely to get neurologic disease than if you're 18 years old and you get infected with West Nile virus. So there's something to do with the host that determines how it's going to be. And if you're immune suppressed, that's really bad. So when people get organ transplants and they accidentally get West Nile virus from their donor, and that does happen, they tend to do very badly and they often die because they're immune suppressed and can't fight the virus. So what we see clinically is that, so Marie-Hélène Théry was a postdoc and now a scientist who did the work. So she's French, so this is in French. But this is form clinique is clinical forms, and then the asymptomatic. So 80% of people don't know they're infected. And we know this from the blood donors because we screen for West Nile virus RNA in the arm, and people are infected. They have no idea. And then I'll tell you a little more about our research, but we find out that a lot of them don't have any symptoms. And then we have the clinical forms, and that's West Nile virus fever. And I'll tell you a little bit more about that in the next slide, but you think of it as a non-specific, you feel like that you have the flu and it's not that big a deal. In fact, it is a little bigger deal than you might think. And then only 1% get these neurologic disease, which is either meningitis, meningoencephalitis, encephalitis, or acute flaccid paralysis like polio, so really paralysis of one limb. And so I think what's remarkable about this is how long the symptoms last. So people who have West Nile virus fever, if you look here, greater than 90 days of symptoms in 20% of the people who get infected. So a fifth of the people have symptoms that last over 3 months. And in fact, if you look at 31 to 90, so greater than a month's worth of symptoms with this viral infection, over half of people. So it's pretty severe. And in fact, the head of the arbovirus branch at the CDC is in Fort Collins, and he got infected with it. Lyle Peterson, call him West Nile Lyle, and he said it was horrible. He was really out for a couple of weeks, couldn't work, and he's not a wimpy guy at all. And he said he felt terrible for months, really, before he was back to full strength. So West Nile virus fever is an entity that's worth looking into. And in terms of the encephalitis, this is in 2011. So when the disease was really ripping across the country in 2003, 2004, we were seeing thousands of cases of encephalitis due to West Nile virus, which is about on par with what we see in enterovirus, a really common cause of aseptic meningitis. And so we thought by 2011, maybe all the bird reservoir had depleted and they were now immune, so we weren't going to see this. It was really petering out the activity. But then lo and behold, in 2012, we had a major recrudescence of West Nile virus activity. And you can see this in the CDC maps as the virus marched across. It hit the Ohio River Valley here and just really likes it there, but then came across to California in 2004. And then you can see that the activity really decreased in 2011, then boom, 2012, there's a lot. And that's summarized here. You can see this uptick in cases. And then last year, the data are out. They don't have the fancy map yet, but about half as many cases were seen in 2013 as 2012. So we're definitely seeing it's endemic in the US now, and it's here to stay. And for some reason that we don't completely understand, it does come back in cycles. And it has to do with the birds and the mosquitoes and global warming and all that stuff. So what we're interested in looking at is why do people get sick? So why does that 20% get West Nile fever? The 1%, we're kind of aware that it's older people and people with depressed immune systems. So you certainly need enough of an immune response. But what is it if you get infected that makes you feel terrible with West Nile virus versus not? And we see this with other viruses as well. If you think of just the common cold, some people have one sneeze and that's their cold, and other people are sick for a week and feel terrible. And that's really mostly our immune response to the virus that determines that, I think. So we looked at a number of different things that might be in play in terms of determining who gets sick. And this is where being at the blood bank is useful. This is a map from the CDC just showing where the infected blood donors are in the US, and you can see that they're scattered all over. But this is sort of the region where we get a lot of infected donors. And at our blood bank, we collect about 1 million units of blood a year. And we test them. And you could never do a research project where you say, I'm going to screen 1 million people a year and find 30 positives and then do research on them. So because we're sitting in the blood supply, we're able to dip our hands in and get those 30 positive donors from around the country, because we test about 6.5 million units of blood a year from around the country. So what we do when we get a donor who's positive, and this is kind of the pain of doing human research, you can't just buy the mice and breed them up. You have to actually find the people, and we send a box out to North Dakota where they did a mobile blood drive, and then it's sent back to us. A lot of coordination, and the blood bank is good at that. And the donors are the best. They already have that altruism gene because they're giving blood already, right? So you ask them, can we have you come in and donate blood every week and then every couple of weeks? They say, oh yeah, no problem. So, so it's really a great group of people to work with in terms of donors. And then we'll find what we do is we have them come in and we give them a phone call and say, do you have any symptoms? We see, fever and stiff neck and all that kind of stuff. And what was really important was the first year we did this, the assay wasn't very good, so we had a lot of false positives. So if you call someone and say you're positive for West Nile virus, how do you feel? A lot of them say, not that great. So we're able to find out, what was true positive versus false positives. And we said if people had 4 or more of these 12 symptoms, it was probably the real thing by comparing our false positives with true positives. So we give these questionnaires at enrollment and then downstream, and we're able to find who's symptomatic and who's not. And then we started looking at things. So the first thing we did back in 2008 was looked at cytokine profiles. So we just had plasma from these donors and said, well, what's going on? And we see that there's mostly a pro-inflammatory response early. So this is before they seroconvert to IgM. So this is early in infection. And then this is after IgM seroconversion. And these are some normal controls. We see a lot of things were elevated, in particular these chemokines, CCL2, CXCL9, CXCL10. And I'd like to draw your attention to CXCL10 or IP10. And we see this in a lot of viral infections. It's the biggest one that's elevated. So in HIV, it correlates most closely with the level of viremia in a subject. We see it in HCV, we see it in HTLV. So I think this, in terms of immunology, is really the viral sensing, or somehow it's most closely tied to viruses being around. But we do see a sort of broad range of immune activation early on. But what we really want to know this is interesting, sort of natural history of West Nile, but does it predict who's going to get sick and who's not? So the first studies we did, we really couldn't see a difference. We didn't have very good granular detail, but more recently we've been able to get samples pretty early on. So right from the blood donation itself, and then subsequent week 1, week 3, and downstream. And we see that the red is symptomatic here. So people who go on to get symptomatic West Nile virus infection, when they donate blood, they're feeling fine, but then they get sick, a few days later. They tend to have higher levels of things like IP10, MCP1, which is CCL2, GCSF. Most of these are not significant if you correct for multiple comparisons, because we looked at 39 different cytokines. A few of them are, and there are a few that are higher in the asymptomatic folks. So there's a hint of a signal here. I don't think this is the answer, but it looks like there is a little bit more cytokine secretion early on in infection in the people who had symptomatic disease. So that's one thing, looking at the innate immune system. What about antibody responses or T cell responses? You could think if you had a more powerful T cell response, maybe you get rid of the virus quickly and there's nothing that you don't get sick. So we looked at that, and the first thing we did was we had overlapping peptides that spanned the whole West Nile virus proteome. And what we found was that the responses are not that high level. So if I come from the HIV field, and we see much higher level of cells secreting interferon gamma. So this is where you can look at how many cells secrete interferon gamma after you stimulate with peptides. And I'd say the responses are not that strong, but they're there. And they're targeting most frequently the membrane, envelope, NS3, NS4A, and NS4B. And if we go through and look, there were some frequently recognized epitopes within envelope, membrane, and then these were recognized by less than 10% of each of the participants. So there were some areas of the virus that were highly recognized, but nothing to write home about. We did do more analysis and found that they were mostly cytolytic CD8 T cells that had granzyme and perforin and looked like they'd be effector T cells. So not a knock your socks off immune response, but then the question is, we know who goes on to get symptoms and who doesn't, so is there a difference? And when we look at that, there really is there's not a difference in the number of peptides recognized by people who are symptomatic versus asymptomatic, or in their total magnitude of response. The one thing we saw was that there were more people in the asymptomatic group who looked like they made no T cell response against West Nile virus that we could detect. So that was maybe there's a slightly stronger response, but nothing to write home about in the symptomatic patients. And then we looked at antibody responses, and there were these are beautiful curves of the evolution of IgM and IgG in an acute viral infection, but certainly no difference between the asymptomatic and symptomatic subjects. So at the first step, it looks like the effector B and T cell responses aren't different between people who develop symptomatic versus asymptomatic disease. So the next thing we wanted to look at was a thought we had. These people are making a T cell response and a B cell response, and they're all just normal people. maybe there's something different about how their immune system is tuned. So maybe there's a Treg tone that when you get infected determines are you going to become symptomatic or not. So Treg cells will damp down the immune response, and maybe a lot of that bystander damage that happens and makes you feel terrible is due to inadequate Treg cells. So we looked at that, and this was a surprise. So these are normal donors here, and they had about 1.5% of their cells were Treg cells. And then if you look at our subjects when we got the first sample we had available where we could get cells was a couple of weeks after infection, because the original blood donation we would have the plasma but not all the cells. They weren't separated out because we didn't know they were infected yet. And then they would come in subsequently at 1, 2, 3 months after infection. Then we had at a year they would come back. And you can see by 1 year, this is sort of like a baseline, a post-infection baseline. They're really not different from the West Nile virus lab donors. These are just folks in the lab who are not infected. But what's remarkable is over these 3 months, it continues to rise over 3 months. This is an acute viral infection. And while some people have persistent symptoms, most of them resolve earlier. And if you look at the plasma, and I'll show you some data on this later, the virus is cleared from the plasma within a couple of weeks. It's not a high-level viremia. So we were surprised to see this long-term perturbation of almost doubling of the regulatory T cell response over time. So that was the first surprise. And then the second thing, which was really interesting, was we looked in those who were symptomatic versus those who were asymptomatic. And our hypothesis looked like it may have been correct, in that the people who are asymptomatic from the earliest time point we could measure all the way through had higher levels of these regulatory T cells. So it may be that the regulatory T cells cause the asymptomatic state, but in humans, we don't have the pre-infection time point, so we can't say that their Treg levels at baseline predicted their outcome. And that, the journal editors really don't like that. They say this is a nice correlation, go to the next journal. So we did get some mouse data to go with this, and I'll show you that in a bit. But one of the things we found curious, I think I alluded to it here, is these Tregs are still perturbed 3 months into the illness. So why was that? And if you look at plasma, you can see the virus goes away pretty quickly in most of the subjects, and most of the subjects are here on this axis are undetectable. But when we look at blood, in the blood compartment, so looking at whole blood, you have to bind up the hemoglobin to get it out of there because it messes up the PCR assay. But if you do that, get rid of the hemoglobin, we actually find that there's virus sticks around for a long time. It's probably neutralized and not active virus, but the immune system is seeing it for quite a while. And if we go down and we look at the plasma or the PBMCs that are separated, it's not in the PBMCs. It looks like it's probably sticking to the red cells. So the virus really persists for quite a while in the bloodstream and probably can stimulate the immune system. So that was interesting to us. And for blood banking, this has implications. If you want to test for West Nile virus, it might be better. We typically test for viruses in the plasma compartment, but really, I think a lot of viruses are sticking around more in the cellular compartment. So we need to figure out ways to get rid of the hemoglobin so we can do more sensitive testing to screen the blood. Then the next thing we wanted to know is, okay, we can find all this virus now in the bloodstream, in the whole blood samples. Do people who have symptoms have more virus? And so we looked at that, and you can see the black bars are the symptomatic people. It looks like later on they had slightly more virus detected. And you can see that by GE analysis, that was marginally significant, that there's slightly more virus in the symptomatic folks. So not a home run. It's not like if you have a lot of virus, you're symptomatic, and if you don't, you're not. But again, this is a small signal that we're seeing. Okay. So getting back to the mouse models, because the journals made us, and I called up Mike Diamond, who's a great guy and did amazing work with us on this. He's a really great collaborator if anybody gets a chance to work with him. And so he has a mouse model where he infects these C57 black 6 mice, and about half of them die, which is a lot more than we see in pupil. And of those who don't die, like at this time point here at 8 days, they can be symptomatic and they get hunched up. And they lose weight. And if we look at the symptomatic mice who are all hunched up, they have more virus than the asymptomatic mice. So in mice, if they're symptomatic and destined to die, they have more virus in their spinal cord and their brain. Okay, why is that important? Well, we then looked at the Treg levels, and the pattern isn't quite the same. As you can see, they don't have the massive Treg expansion that we saw in humans, but those mice who did survive and had the lower viral loads had higher Treg levels than those who did not survive. And if you look at the level of T cell responses, if you had high West Nile virus specific T cells, so a bigger T cell response, which we almost saw in the humans, but very weak, they had lower levels of Treg. So the data were consistent in the mice. And if we knocked it out using a DTR knockout mouse, we got some from Sasha Rudensky's lab, this is a diphtheria toxin floxed in. So if you give diphtheria toxin, it wipes out all of the Treg cells that have FOXP3, and those mice all die, and the other mice have normal mortality, even if you give them just DT and they don't have that FOXP3 knockout. So with an inducible knockout, we can say that the Treg cells in the mice are really required to help protect against lethal infection. And in humans, what we see is if you have higher Treg levels, then you're somewhat protected from symptomatic infection. And then some of our more recent work is looking at the quality of the adaptive immune response. So we know that just interferon gamma secretion by these T cells doesn't seem to differentiate people who go on to get symptomatic disease from those who don't. But we looked at a couple of things. There's T cell immunoglobulin domain, mucin domain 3, which is a big mouthful. We call it TIM3, and PD1, programmed death receptor 1. And these molecules have been described on the surface of T cells during chronic infections like HIV as being associated with exhausted T cells that really can't function all the way anymore. But I think what we're learning is during acute viral infections, these probably are markers, especially TIM3, probably a marker of an activated T cell. So we then looked in West Nile virus infection. We found, compared to normal controls, which is this little dot here, that our symptomatic subjects with West Nile virus infection had a lot higher levels of TIM3 positive T cells. So while the interferon gamma secretion was about the same, whether you had symptomatic or asymptomatic disease, you actually have more activated T cells and you have fewer regulatory T cells if you're a symptomatic patient. So we are learning a little bit about the disease, and PD1 levels were not different. The other thing we looked at was markers of senescence. So if you have older T cells, they have this marker CD57 on them. And those levels were basically, the difference we saw in TIM3 levels were in non-senescent cells that did not express CD57. So the CD57 positive cells had no difference in the levels of TIM3 on them. So it was really in these younger effector cells that we see they're activated and have TIM3 on them. And then finally, I think this may be the last data slide. Galectin-9 is the ligand for TIM3. And what it does is if a cell has TIM3 on its surface, galectin-9, it's a protein, comes along, binds to it, or it's a lectin, it binds to it and it causes that T cell to apoptose. So it's a way of negatively regulating these T cells that are activated and perhaps dysfunctional in chronic infection that have TIM3 on their surface. So when you dumped galectin-9 on it, it gets rid of them. So we looked in our humans who were infected, and we found that compared to normal controls, people with acute West Nile virus infection have an upregulation of galectin-9, which is this negative regulatory molecule. So we say, well, this all looks consistent. So maybe the asymptomatic folks will have even higher levels of that negative regulatory molecule, and that will explain things. So we went into the mice. Went back to Mike Diamond, and he had some galectin knockout mice. And if you knock out galectin-9, they die more. So again, they're missing a negative regulatory thing, just like the Treg deficient mice. If you infect them, they get too much inflammation and they die. And in fact, they have higher viral loads, just like the Treg knockout mice. And the frequency of their T cells, they actually have a more vigorous CD8 T cell response, again, just like the symptomatic mice who are Treg depleted. So if you're missing that negative regulatory factor, you do worse if you're a mouse. Unfortunately, when we looked in the humans, there's a trend for the asymptomatic folks to have more of this galectin-9 in their serum, but it didn't reach statistical significance. So this doesn't look like a complete answer, but maybe a partial answer. So now I've showed you a lot of different things. I mean, this was maybe 4 or 5 papers we just buzzed through right here. So how do we put it all together? I would say West Nile virus induces an acute inflammatory cytokine response. We saw that, especially IP10, some of these chemokines. The T cell response is nothing to write home about. It's there, but it's not particularly strong, and it's not particularly different between the symptomatic and asymptomatic. Treg cells really double over this acute infection. That's a surprise, and that hadn't really been shown in a viral infection in humans before. Treg cells have been looked at a lot in autoimmune diseases and cancers, but not so much in viral infections. And then the TIM3 positive cells and galectin-9, this sort of yin and yang, they both rise during acute infection as well. But the real question was what determines who's symptomatic and who's asymptomatic. And what we have there is that cytokine responses are a little more vigorous in symptomatic infection. We have a lot of these where they're just a little bit different between the two. Nothing's a home run. The effector T and B cell responses are pretty similar in the symptomatic and asymptomatic subjects. Maybe in the mice we see a more vigorous T cell response in the symptomatic mice that have higher viral loads. Treg cell levels are higher in asymptomatic subjects. This was the thing that stood out the most. This was the clearest difference, that they had a higher Treg tone. The viral load is just a wee bit higher in the symptomatic infection, and TIM3 levels are higher in symptomatic West Nile virus infection. So can we put all this together with a model? So what happens, and the answer is no, we can't yet, but I have a model anyway. So you get West Nile virus infection happens, you get some inflammation, we see all these cytokines going up, you activate your T cells and they upregulate TIM3 and galectin-9 tries to downregulate these cells, and then you have some antiviral T cell activity. So what happens in asymptomatic disease is that you have maybe a little less virus happening, maybe you have a little better antiviral activity, not clear on that. And we do know that we have higher Tregs that damps down this inflammation, so maybe that makes the cycle better. And with symptomatic disease, you have more inflammation, a little bit more virus, and this is where we really don't know. Do these T cells not function as well? We haven't answered that question yet, but that's one hypothesis. It's really not clear to me why the symptomatic disease has this cycle of inflammation, and that's something we need to work on still. But this clearly happens, I think that we know that. So with that, I'll close and thank, this is Marion Lanterie. She's really done all of the work. I'm just here talking. Mike Bush started the program at Blood Systems Research Institute. We've had samples from the Red Cross. Mike Diamond and his crew, I've mentioned, have been great collaborators in the mouse work. We got Treg knockouts from Jenny and Sasha, and then funding. So with that, thank you.
Thank you. So it is unusual to find more inflammation and more virus when you expect that if you have a bigger T cell response, you get less virus. You might get more death because you get an overshooting inflammation. You get pathology induced by the overshooting immune response. But what you're showing is more T cell response is actually associated with less virus. So I'm wondering whether what you're actually seeing is more antigen means more T cell responses, and that the virus replication is really not controlled by the T cells, but by some other factor, probably innate.
Yeah, that's a really good question. So the other thing that we haven't discussed in detail is that this, in the mice at least, this is really getting through the blood-brain barrier. So that's also an important part of the puzzle. The T cell responses aren't a whole lot higher in the humans if they're symptomatic versus asymptomatic, but in the mice there are more T cells, and I agree that I think antigen is probably driving that. And for some reason, those T cells, even though there are more of them, aren't effective in controlling the virus in those mice. And part of it, maybe the inflammation is breaking down the blood-brain barrier more, and so they're getting into the CNS. But that's an active area of research, is trying to figure out what elements, it may not be the T cells at all, as you mentioned, it could be the innate immune system. We've done some work with Michael Gale up at University of Washington looking at IL-1 beta as being important in activating the inflammasome, and if you knock that out, the mice do a lot worse. The thing about the mice studies is if you knock out any arm of the immune system, they do worse. You know, you knock out the CD4 cells or so it's sometimes hard to tell what's the key part to put it all together. Good question.
There we go. I might show my naivety here a little bit.
Yes, there.
But is the your 20% of symptomatic patients analogous to what you would see in dengue virus? Is there a possibility that they're infected with another West Nile-like virus?
So do you think that they're infected with like dengue as well as West Nile? Is that what you're saying?
Not necessarily dengue, just another virus that you get an increased inflammatory response to West Nile virus-like antigens.
Yeah, no, I think it is just West Nile virus that's in these. We haven't done sequencing to make sure there are no other viruses, but we know that they have an acute infection, and we see them seroconvert. And you bring up dengue, and in fact, with the Treg data, Sarah Rowland Jones from Oxford looked in people who had severe dengue versus asymptomatic dengue in kids, and she found that the Treg levels were slightly higher in the folks who had asymptomatic infection compared to severe dengue virus infection. That was in the Gambia. So I think that the data are consistent with other viral infections. And then in mouse models, Sasha Rudensky's group looked at HSV and LCMV and found this same thing with the paradoxical higher virus load in those who had the lower Treg levels. So it's not entirely, at one level you'd think that having higher Tregs would mean your T cells wouldn't function as much, so you get more virus, but in fact we see the opposite. And no one's really given a satisfactory explanation for that, though it's been seen in multiple labs.
Hi, Phil.
Yes.
Have you, hey.
Hello, Shomi.
Have you considered the HLA genotype of your patients who are symptomatic versus non-symptomatic? Have you guys tried to group those at all?
Yeah, good question. We actually have a publication on that. I just didn't put every publication in here. So we looked at the HLA type and tried to see was there an overrepresentation of one HLA type versus another. And we didn't really come up with much. We found some weak associations, but that didn't seem to be the home run as the HLA background. Other people have looked at genetic screens. So Mark Loeb up at McMaster University had a massive contract, and they looked at people who were hospitalized who had severe West Nile virus versus asymptomatic, and they really didn't find much in terms of genetic. There have been a few people. Margot Brenton has found OAS genes that seem to be predictive. CCR5, well, it's great for HIV not to have the CCR5 delta 32. You're actually more susceptible to West Nile virus. There have been a couple of publications showing that if you lack CCR5, you're more susceptible to this virus, which is surprising because we thought there was really no effect from our days at looking at HIV-infected patients, but there probably is a reason to have that receptor. Maybe last two questions, then we'll go on to the next speaker to cut myself off.
So my question is regarding the specificity. So as you know, there's wide across immunological reaction among different flaviviruses, including St. Louis, JEV, dengue, and West Nile virus. Okay, and when we performed serological testing, we did notice some kind of confusion between different flavi, and sometimes we have to perform PINT to do the differentiation diagnostic between different flaviviruses. For your, because it is a T cell mediated immunity. So did you notice any specificity? I mean, my question is whether your research here is specific for West Nile virus, or have you tried any other flavivirus for your
Yeah, no,
experiment?
that's an excellent question. So, as the questioner pointed out, these flaviviruses are fairly closely reactive. So when we see IgM come up and IgG, would that be someone who had received a vaccine for Japanese encephalitis virus, or had been down to South America and got dengue virus in the past? So we actually did that. We sent these samples to the CDC for PRNT analysis, and there were, as I remember, out of the cohort that I showed you with the Treg studies, maybe out of 35, I think 4 had seen, 1 had a JEV vaccine, and maybe 3 had dengue in the past. But they didn't segregate into the symptomatic versus asymptomatic group. So it wasn't that they had a pre-existing immune response that made them either protected or more susceptible. So there was no difference in the groups in the distribution. People had some prior flavivirus activity. Okay, one last question.
You've demonstrated the great power of being able to screen 1 million+ individuals. Given that power, how do you decide which targets to focus on, and how can other researchers gain access to the resource that you have at the blood bank?
Yeah, no, excellent question. We work with a lot of other groups, and as long as we have the samples, we say yes. So we work on what interests us and what we think is the next big thing in immunology, and we may or may not be good at that. But we also work with a number of other groups, and so some of the samples are part of an NIH repository because it's funded by one of these ARA awards. And so that has to be applied for through a formal mechanism, BioLink at the NIH. The rest of them, people just contact us and we share our specimens and we write grants with people to get more of these specimens because it is expensive. But we're happy to collaborate. So if you want some, come see me at the break, or anybody.